Malone. I'm going to invite up Peter Schneider, who's now going to talk us about will the drug-coated balloon and drug-eluting stent have a future in lower extremity revascularization, an update after the Paclitaxel summit at the VLF.
Thank you, Dr. Leyden, Chairman, and esteemed panel. So I would like to spend the next few minutes addressing our most favorite subject of all time. When Paclitaxel becomes a household word, you know you have a problem on your hands. And these are my disclosures. So I would just purport that what doesn't kill you makes you stronger. And I think that this whole Paclitaxel issue, it does have the potential to make our field stronger, more mature, and better than it's ever been before. So this is a test case. It's a crisis of uncertain magnitude and etiology, but dramatic negative effects if we choose the wrong path. The meta-analysis that kicked this off asserted an increased risk of death, and that's an attention grabber. And of course, with the presence of a dose relationship suggesting causality. So what did we learn so far, which is what I'd like to cover in the next few minutes, about trial design, drug delivery, vital statistics, and resilience of our own field. The roles of the key participants, the FDA, international regulatory bodies, societies, study participants, vascular practitioners, and of course, we've heard less from the patients, but keep in mind that the patients are the recipients of these complex pre-treatment discussions that we're having in the office. And of course, what are the next steps? So we know about the efficacy of Paclitaxel. Here are three separate randomized controlled pivotal trials looking at 12-month patency, each one showing a significant benefit. So what we see here is sort of the one-year patency gridded against lesion length, which we know the patency decreases over the longer lesions. So this is PTA, and these red dots are stents. So these are the task A and B stent versus PTA trials of the past 10 to 15 years. And when you look at DCB, DES, what you see is superior performance in the pivotal trials, and now even some registry data over the past couple of years looking at longer lesions, significant task D and above, with appreciable patencies that seem to be breaking this mold of worse patencies with longer lesions. This comes from the FDA panel pack looking at the pivotal trials that were studied, that were done primarily within the US and were done for approval, and looking at efficacy. This is clinically driven TLR at 2 years, showing a relative risk of 0.57. That is a benefit for Paclitaxel. And a more recent meta-analysis from the group in France showing a CDTLR at 2 years with a relative risk of 0.29. That included some of the randomized trials from around the world. So along comes a meta-analysis at 2 years, hazard ratio 1.68, 4 to 5 years 1.93. We know those numbers. And we see that drug-coated balloons and drug-eluting stents had been about, in the latter part of 2018, up to 50% or a little bit more of the cases. And then, of course, a significant decrease down to about half of that over the next 2 quarters. So the characteristics of these randomized trials that we're talking about, they were powered for one-year patency, not for long-term mortality. And I have to stress the importance of that because you're going to get the answer to the question you ask properly. And we weren't really asking for the answer to the long-term mortality question. Small control groups, some of these randomized trials were 2-to-1 randomization, means unstable estimates. And the question is there ascertainment bias in the assessment of mortality? In other words, meaning that although we randomized them up front, did we assess the mortality difference differently in the long run? And then, of course, were both groups treated the same? Again, randomized up front, matched in so many ways, and yet were both groups treated, that is with medical treatment, the same way over the long run? And then other questions that have come up. Is there a dose response, that is a biological gradient? Is there a clustering of deaths as to cause, suggesting mechanism? Was there a consistent signal, and is it a causal relationship or an association? Of course, the Bradford-Hill criteria, which were initially published in 1965, really assess the degree to which causation between a specific cause and an event are associated, and they rely on these, dose response, mechanism, etc. Well, the first and easiest to tackle is the dose. So the initial meta-analysis that suggested a dose relationship, again, a biological gradient, assumed, because it included time, assumed a continuous linear and increasing exposure over time. So we know from preclinical models that the paclitaxel in tissue levels is almost undetectable at 6 months. And time is disproportionately available for studies with longer-term follow-up. And these were the 5-year studies that we're talking about. And my layman's sort of practitioner's non-statistical way of looking at this is that the longer you follow someone and the older they are, the more likely they are to have a mortality event. That doesn't seem like rocket science to me. In any event, so the dose effect on mortality, this comes from the FDA panel pack, and although the up and down axis is the same in each one, you can see that the dose is different because these were different dosages on these different devices. And there was no mortality, clear-cut mortality effect. And here's another study with 1,837 DCB patients using a specific and single preparation, showing, again, fairly tight curves between the different dose terciles. This again comes from the FDA looking at different devices with the dose delivered and the relative mortality risk ratio. And actually the one with the higher dose had the little bit lower mortality risk ratio. Here's another study, again looking at cumulative incidence of all-cause death with paclitaxel divided into terciles, with actually the lowest tercile having the highest mortality. And of course the p-value 0.73. So in the FDA letter from August 7, quoting directly, no clear evidence of paclitaxel dose effect on mortality, no identified pathophysiologic mechanism for late death. So the FDA is in the middle. They're between a rock and a hard place. They need to serve the public health, not one group or another. So the other thing about the dose effect that really never made sense to me is that we know that the dose for coronary paclitaxel use is quite small. And we don't use that. I'm not a coronary practitioner, but we don't use that anymore because we have better DES, more effective. The dose that we're talking about is this middle range, and the dose for cancer, total dose, typically 1,100 to 1,500 milligrams for intravenous paclitaxel treatment for the total course of therapy. So we're just talking about it for this middle dose, which again doesn't quite make sense. So no clustering of deaths. What you see in the blue bars is paclitaxel, red bars is non-paclitaxel, and you can see that both for CV death of different types and non-CV death, there were more deaths attributed to the group that got paclitaxel, but there was no clear specific cluster. And that really questions the whole issue of biological mechanism. If you go a little further and you look, okay, what about the missing data? Well, you can see that the missing data pre-FDA panel and the tremendous amount of work that all of the industry sponsors did to track down the missing data at a later time showed that a substantial portion of the missing patients were located and cause was ascertained. 20 to 86% of missing patients were found, and you can see the biggest jump was in the impact SFA. The 5-year point estimates from FDA, relative risk 1.72, but after ascertainment of some of those missing patients, it was down to 1.57, a decrease of 21%. And in fact, in the group that had the highest degree of ascertainment, the decrease was a relative risk of 1.63 down to 1.39, a decrease of 38%, suggesting there was ascertainment bias. In other words, they were randomized going in, but not randomized going out. They were lost at a different rate. There was a geographic variation with a bigger difference in the mortality curves in the U.S. than outside the U.S. in these two major 5-year trials. In Japan, there was no difference, again, suggesting that if it's going to be dangerous, it should be consistently dangerous. One potential source of answer to that question is the 5-year, or let's just say over the years, the attendance of follow-up visits was much better in the PTA group than it was in the DCB group. And although this crossed regions, it was largest in the US, where the signal was the largest. So again, suggesting treatment bias. And this is probably the most important slide, and the slide that I'll end with. Let me just say, here's the initial meta-analysis, 1.93. FDA pre-vital statistic, 1.72 down to 1.57. The VIVA independent patient analysis will be discussed in great detail tomorrow by Krishna Rocha-Singh. Look at all these data bits that have come up since the FDA panel, the VQI, the Optum database, the Medicare inpatient and outpatient comprising 152,000 patients, and then these other data bits, none of them actually showing an increased mortality for paclitaxel. So I think, given this, and of course this is the German database from 2 weeks ago, the German health insurance system showing showing no signal for paclitaxel. So I think the bottom line is that a lot has happened since this was first identified. The participation of the FDA and the collaboration with which they have gone forward has been tremendous. The ability of the societies, the different physician groups, and all to come together gives me great hope for the sophistication of our field going forward. Thank you.